Hi everybody, it's your human anatomy and physiology teacher, Mr. Poser. Today we are going to be discussing the really nitty-gritty components of how a muscle is able to contract. So we're going to be getting down to the molecular level and discussing how one muscle contracts. So a few things I want to recap before we get going. Really all muscles do, all they do is contract. Okay, and if you remember the structure of a muscle is made up of filaments, which make up a fiber, which is a muscle cell, and the fibers are bundled together and wrapped in connective tissue, and then a bunch of bundles are all bundled together, so it's just bundles and bundles and bundles and bundles of fibers, right? And all a muscle does is contract. So all those filaments and fibers, all they're really able to do is just make themselves shorter. And every single motion that's done within your body, besides maybe a few self-propelled, cells, it's really done by the muscular system or some kind of muscle. All right, and that's all it really does is contract. So we're going to get into how is a muscle able to contract today. Okay, but so this is where we, I believe, left off in the last video. How a muscle is able to even start doing anything, a muscle has to be stimulated first. It has to be told what to do most of the time. Well, the skeletal muscle. Smooth muscle has their own kind of system, but that's for another day. Muscles need to be stimulated, okay? And what we filled out last time, a diagram that we filled out last time is the sarcomere, okay? And that is the smallest unit within a muscle fiber that carries out contractions, okay? So we labeled this last time. All a muscle cell is really trying to do, let me do something really quick, sorry, hold on. All a muscle cell is really trying to do is bring the Z discs closer to the H zone. That's all that's happening. All it's trying to do is just go zoop like that. Okay, and if you look closely here, okay, you have two different types of filaments that we already identified on the diagram. We have actin and we have myosin, and those are what we call myofilaments. And those molecules are specialized proteins that are going to pull on each other. So they're kind of arranged like this. You see my hands right here. They're kind of arranged like that. And all they do is they slide on each other like that, and they glide and they pull on each other and they slide past like another like so. That's called the sliding filament model. That's our understanding of how a muscle contracts. Okay, so let's keep looking here. A couple other things that we need to know before we get into the steps of muscle contraction is that they are complex and they're involved in influx of calcium. Okay, so calcium ions are going to be kind of like these signaling molecules that cause a change within a muscle cell, and that muscle cell is going to be triggered to contract. Okay, so calcium ions, where is that stored within the body? Well, it's stored within the skeletal system, as we learned in our last unit. So that's another way that the muscular system and the skeletal system are linked. Okay, muscle pulls on bone, but bone stores the calcium for muscles to contract. So it's pretty helpful. And they need a lot of ATP. Okay, if you remember from way back in the beginning of the year, we discussed ATP. ATP is the main energy molecule for all living things. All right, it's what we call the cellular energy currency. Your mitochondria is the powerhouse of the cell, are going to be making ATP at a really high rate within muscle cells because your muscles need a lot of it. In fact, I want to say like 40% of your daily ATP usage is just from your muscles. And think about it, some of them always have to be contracting in order for you to not be this like glob. Okay, so ATP and ADP, okay, we remove what we call an inorganic phosphate from ATP and it becomes ADP, but we use that energy that from the bond that held that phosphate in. Okay, so muscle movement is caused when actin slides over myosin. Actin, myosin, they slide past one another and cause those Z lines to contract to get closer to one another. All right, this is for a model that we're going to be doing later in class. Okay, we're going to be acting this out. It'll be pretty fun. But muscle contraction occurs via what we call the sliding filament model. That's our understanding, our best explanation for how muscles are able to contract. And it's very well researched. So even though we call it a model, it's very well researched and it's how a muscle contracts. Okay. All right. So before any of those filaments start to slide or anything like that, okay, a muscle, particularly a skeletal muscle, because that's what we're focusing on, it needs to be told what to do by the nervous system. So that is where the neuromuscular junction comes in. So let's break apart the word neuromuscular. Neuro is referring to a neuron or to the nervous system, and muscular is referring to, well, muscle, right? So a nerve impulse, because nerves communicate with each other through impulses, electrical impulses called action potentials. This is going to be a topic that we get into next semester. Reaches the end of a nerve cell and it causes it to release a compound called acetylcholine, ACH. All right, and acetylcholine is what we call a neurotransmitter, meaning that it's taking a message from a neuron to another cell, whether that be a neuron or in this case a muscle cell. Okay, so as I drew here, there's an arrow, there's an electrical signal that passes from the neuron, okay, and then these vesicles, well, these little bubbles of cell membrane holding this neurotransmitter at the bottom of the neuron, it's called the axon terminal, okay, their job is to release this blue chemical, it's called acetylcholine, into what we call this synaptic gap. Okay, this gap between the nerve cell and the muscle cell. And see these little goalpost looking things? These are representing what we call receptors. Okay, so receptors are special proteins on the membrane of the cell that are going to pick up that signal from the neuron. Okay, and when the signal binds to the receptor, okay, it's going to cause a fundamental change within that neuron. Okay, so basically what we're looking at is how does the message from the nervous system get to the muscle cell. Okay, once again, electrical signal passes through here, causes the cell to release a chemical which is picked up as a signal by the muscle cell, and that is going to cause a change within the muscle cell. And what is that change exactly? Well, I'm getting a little ahead. ACH, that acetylcholine, that signaling molecule, sends an impulse, sends another electrical signal caught down something called T-tubules into the sarcoplasmic reticulum of the muscle cell, causing it to release calcium. All right, so we talked a little bit about the sarcoplasmic reticulum in our last video. It's like the endoplasmic reticulum of a regular cell, okay, but since it's a muscle cell, it's the sarcoplasmic reticulum. So we're just going to call it the SR. Okay, so ACH, acetylcholine, it's a signaling molecule. It basically causes the release of calcium into the muscle cell. Okay, so this is where calcium is coming in. We hinted at that before. It receives that signal and it causes the muscle cell to release calcium. Okay, hey, release some calcium. That's what's happened so far. All right, and what does that calcium do? Well, that calcium is going to cause some other changes that are going to allow the muscle to contract. Okay, so I have two pictures here. Well, it's one picture, but it's a before and after. Okay, so it says calcium ions bond to troponin, shifting the troponin-tropomyosin complex and exposing the binding sites on actin. Now, what the heck does that mean? All right, now we have a really, really close-up picture of actin. Okay, and actin is one of the two filaments that are going to be sliding past one another. All right, so if we go all the way back to our sarcomere, okay, actin are these thin blue lines. Okay, so we're getting really, really small here to the molecular level to figure out how muscles contract. Okay, so here's actin, and in order for the muscle to contract, actin and myosin need to pull on each other and slide past one another. But here's the thing, when the muscle is relaxed, when it's not stimulated, it's got two molecules, two protein complexes that are going to be blocking that actin and not allowing myosin, the other filament molecule, to bind to it, and they're not going to allow them to slide. Okay, so with these molecules in the way, troponin and tropomyosin, okay, these fibers, these filaments, I should say, are not going to slide. They're not going to cause the muscle to contract because they can't pull on each other. Okay, that's until calcium comes in. All right, so remember, we released calcium as a result of receiving that signal, and calcium, what it does, it basically gets troponin and tropomyosin, these two proteins blocking actin, out of the way. It just says, hey, move over, move aside, step aside. And it causes the myosin binding site on the actin filament to be exposed, meaning that myosin can now bind to actin. These two filaments can start to pull on each other and slide past one another and allow the muscle to contract. Okay, so calcium's going to really start things off here. It's going to clear the way for these two filaments to bind to one another. Now, here's our actin, and we got calcium and troponin, tropomyosin out of the way. Okay, now this is where we get into the contraction part. ATP, our good friend, our energy currency molecule, breaks apart into ADP plus P and attaches to the myosin head. All right, so we have a high energy molecule, and what it's going to do, it's going to come over here to this myosin, which is the thick filament, and it's going to cause a conformational change. It's going to cause the myosin to change shape. Okay, here's the thing about myosin. It's got these head-shaped structures. It's like golf club-shaped structures right over here. So I just outlined it like that. And these golf club-shaped structures, these are going to be the parts of that myosin that are going to reach across and bind to actin. Okay, so in order to be able to do that, it needs energy. It needs ATP. So ATP drops off a little bit of energy in step 4. And then step 5, the myosin head, what we call, forms a cross bridge and binds to actin with the ADP and an organic phosphate, the P, still attached. Okay, so we use that energy, and that golf club shaped filament, or the part of the myosin, reaches across and binds to actin. Okay, so if this is actin, this is myosin. Myosin is going to kind of reach across and grab onto actin, okay, with the help of ATP. Now, in order to get it to contract, okay, we need to start pulling. Okay, so what we're at right now, myosin binds to the actin, and it says ADP plus P detaches from the myosin head, causing the myosin head to bend, resulting in the power stroke. You have to say it like that. Okay, so what happens here is that the myosin changes shape. Once again, ADP kind of like detaches, okay, and then what it does, it causes the myosin to change shape and pull on the actin in one direction. Okay, it's almost like a rower, somebody rowing. They go pull like that power stroke on the actin. Okay, so actin gets pulled towards this middle of the sarcomere. Okay, so what starts to happen here on a bigger scale, if we're looking at the sarcomere as a whole, is that, let's just pretend this is myosin and this is actin. Myosin starts to pull on actin, and these start to slide past each other. Okay, so ATP allowed it to form a cross bridge. When it breaks off, when ADP breaks off, it causes the power stroke and causes the proteins to pull on the actin. Okay, from this point, a new ATP molecule binds to the myosin head, causing it to detach from actin and return to the loaded position. So once again, we have an ATP molecule. Thank you, ATP. That allows it. So we pulled on actin. Okay, we pulled on it. Now we have to detach. So ATP is going to come in again. Okay, and it's going to allow the myosin to break off from the actin and return to its original position. Okay. So steps 5 through 8 repeat until calcium is no longer present, causing the troponin and tropomyosin shift back to the original position, which ends contraction. Okay, so what does this all mean? Okay, 5 through 8. Let's go back to 5 through 8. Myosin forms a cross bridge with the help of ATP. ADP detaches, pulls on the actin, causing it to contract, and then the myosin detaches once again thanks to ATP, and the process can start over again until calcium is no longer present. Okay, remember, calcium got rid of this troponin and tropomyosin complex. Okay, and when the muscle is no longer stimulated, it's no longer being told to contract by the nervous system, there's going to be no more calcium. And calcium's, well, once it leaves, I'm going to put some arrows here. Once it leaves, troponin and tropomyosin are free to kind of bind to actin again and prevent myosin from pulling on it anymore. Okay, and that's relaxation of the muscle. Okay. That is where we're going to stop for today. All right, so I'm going to run through it just one more time. Okay, nerve cell sends a signal to the muscle cell. Muscle cell picks up the signal. Okay, the signal causes the muscle cell to release a bunch of calcium. The calcium gets troponin and tropomyosin out of the way, allows myosin and actin to bind to one another. Okay? Myosin, with the help of ATP, forms a cross bridge. It reaches across to the actin and binds to it. When so it reaches across and binds to it. Sorry, I skipped a step there. And when ADP detaches, it results in the power stroke, which causes myosin to pull on actin and pull actin towards the middle of the sarcomere. Okay? And then ATP rebinds to to myosin, pulls off of it, and that process repeats until there's no more calcium and contraction is going to end. All right? That's it for this video. Please, I understand it's complicated. Please let me know if you have any questions, and we will see you in our next video. Bye.